Usually when we talk about a medical diagnosis, there's this expectation of mechanical precision. Like, you break your arm, the X-ray shows that jagged white line, and the doctor just points and says, yep, there it is.
Right. It's very black and white.
Exactly. But then you step into the world of neurodegenerative diseases, and suddenly that X-ray machine feels like it's just, well, completely broken. We're looking at a diagnostic landscape that is incredibly murky, especially when we start comparing conditions like amyotrophic lateral sclerosis, which is ALS, or frontotemporal dementia, and Alzheimer's disease.
Oh, absolutely. Because under a microscope, those brains look like completely different crime scenes. I mean, if a patient suffered from ALS or FTD, their brain cells are typically overflowing with this malfunctioning protein called TDP-43.
Okay, so a specific kind of cellular garbage.
Right. But then you look at an Alzheimer's patient's brain, and you see something else entirely. You see cells just choked by tau tangles and amyloid plaques. So it's different diseases, completely different protein garbage building up inside the cells.
But the end result is the same, right? The neurons die, which kind of brings up the mystery I want you, the listener, to hold in your mind as we get into today's source material.
Yeah, this is a fascinating study.
It really is. It's a groundbreaking study from the journal Cell. And the question it asks is, if the protein garbage is so different, what if those proteins aren't the actual executioner? What if ALS, FTD, and Alzheimer's all share the exact same microscopic killer sitting way deeper in the cell, like right down in the DNA?
And, to find an executioner operating at that level, the researchers behind this study had to look at the brain at the absolute highest possible resolution.
Which means looking at single cells.
Exactly. They couldn't just look at general brain tissue, they needed specifics. So they isolated postmortem brain tissue from patients carrying a really specific genetic mutation called C9orf72.
Let me just pause on that real quick. That specific mutation, that's the most common genetic driver of familial ALS and FTD, right?
Yep, that's the one. So they took that highly specific tissue and set it up in a direct comparison alongside tissue from Alzheimer's patients and also, neurotypical control brains just as a baseline.
Right.
But the critical choice they made was in how they looked at this tissue. They used a technique called single-cell whole-genome sequencing, or SCWGS.
Okay, this is where I have to push back a little bit on the methodology because, wait, why go through the agonizing, incredibly expensive trouble to sequence single individual cells?
It is a lot of work.
Right. I mean, if the goal is to find DNA damage in a diseased brain, why not just take a chunk of the affected cortex, put it in basically a biochemical blender, grind it up, and sequence the whole thing to get a massive, clear signal?
Because if you blend the tissue, you actually destroy the signal completely. The human brain is a mosaic. Think of a crowded stadium where 1 million people are talking, but only 10 people are whispering a vital secret. If you hang one microphone over the whole stadium, you just record white noise. You have to mic up every single individual. In the brain, neurons accumulate DNA mutations independently of each other.
I see. So if you bulk sequence a whole chunk of tissue.
Right. A catastrophic mutation that exists in just one dying neuron gets entirely drowned out by the billions of normal DNA letters from the neighboring healthy cells.
Okay, the stadium analogy makes perfect sense. You need to read the instruction manual of one specific dying cell. But to read a single cell's DNA, you have to amplify it, right? You have to make enough copies of it to even run it through a sequencing machine.
You do, yeah.
So if a cell is already dying and fragile, wouldn't copying its DNA introduce a bunch of, artificial errors?
And that is the exact hurdle that has held this entire field back for years. Standard amplification is like taking a photocopy of a photocopy of a photocopy. Any smudge on the first copy gets exponentially worse.
Right, it just turns into a blurry mess.
Exactly. So to solve this, the researchers used a relatively new method called PTA, which stands for Primary Template-Directed Amplification.
So instead of copying the copies, the chemical machinery always goes back to the original source document.
Yes, it amplifies the original template directly. This suppresses those artificial errors and gives an incredibly pure read of that single cell's genome. So, armed with this, they sorted the isolated ALS and FTD neurons based on whether that toxic TDP-43 protein was physically present or absent in the cell.
The expectation being that the cells overflowing with toxic TDP-43 would obviously have the most mutated DNA.
That is exactly what you would think. But the presence or absence of that specific protein pathology didn't seem to be the primary driver of the DNA damage at all.
Wait, really?
Yeah. The neurons depleted of normal TDP-43 didn't show uniquely higher mutation rates compared to the others. Instead, the finding was much broader. Like all the disease neurons across ALS, FTD, and Alzheimer's simply had massively elevated levels of somatic mutations compared to healthy brains.
Okay, so we need to look closer at what kind of mutations we're talking about because the errors in the paper come in two main flavors. First, we have SSNVs, which stands for somatic single nucleotide variants. And second, we have SINDLs, somatic insertions, and deletions.
And the distinction between those two is really the key to this whole mystery.
The way I picture an SSNV is like a single typo in a word. The cell's instruction manual types cat instead of bat. The sentence might still be readable, or the cell can kind of work around it.
Right, it's a minor glitch.
But in SINDL is entirely different. That is like someone taking a thick black marker and redacting an entire word, or just ripping a whole paragraph out of the book. The information isn't misspelled, it's just permanently gone.
Yeah, and taking that black marker analogy a step further, imagine someone redacting those words hundreds of times over in a single manual. The diseased neurons definitely had an elevated number of those single typos, the SSNVs.
Right.
But the truly staggering finding was the volume of SINDLs. Some of these individual disease cells had over 1,000 somatic insertions and deletions.
Wait, over 1,000. Give me a baseline here. How many missing paragraphs does a normal, healthy brain cell have?
A healthy neuron might accumulate maybe a couple hundred of these deletions over an entire 80-year human lifespan.
Oh, wow. So we are talking about the equivalent of hundreds of years of normal, age-related genetic decay happening all at once in these diseased brains.
Exactly. The manual is just rapidly losing pieces.
Okay, let's pause and stitch this together for a second before we move on. We've got these single cells from different diseases, and regardless whether they are choking on tau or TDP43, their DNA is basically being shredded in the exact same way.
Yes, they're missing massive chunks of code.
So if pages are being ripped out of the manual, how do we know what is ripping them out? Like, are they being deleted at random, or is there some kind of pattern?
They are absolutely not random. They leave a highly specific fingerprint. And to find it, the researchers used a computational approach called de novo mutational signature extraction.
Which sounds like a massive mouthful, but if I understand it correctly, it's basically an algorithm designed to find patterns in huge data sets.
That's exactly right.
You feed the computer thousands of these random-looking DNA deletions, and it just groups them by similarities.
Right. It looks at the DNA letters immediately surrounding the deletion, the exact size of the deletion, and extracts a statistical signature. Once the algorithm defines that signature, researchers can cross-reference it with a massive global database called COSMIC, which is the catalog of somatic mutations in cancer.
Which is such a brilliant resource to tap into. I mean, cancer researchers have spent decades mapping out the exact genetic fingerprints left behind by different toxins, radiation, and broken enzymes.
Exactly. So first, the algorithm analyzed the single-letter typos, the SSNVs. It extracted a signature that perfectly matched a known cosmic profile called SBSB.
And what does that one do?
Well, this specific profile is the universal calling card of oxidative DNA damage.
Ah, okay. So the cells are under intense metabolic stress. They're essentially rusting from the inside out, and that oxidative stress is causing typos.
Right, but the typos are just a symptom of the environment. The real executioner is revealed when the algorithm extracts the signature for the missing chunks, the SINDLs.
The ripped out pages.
Right. The algorithm pulled out a signature they named IDA. And when they ran IDA through the COSMIC database, it matched perfectly with a known signature called ID4.
And this is where the sheer precision of this mechanism comes into focus, because the algorithm revealed that this massive spike in missing DNA wasn't random, jagged chunks being torn out. It was specifically, almost exclusively, 2 BP deletions.
2 base pairs.
Exactly, 2 letters of DNA just being snipped out over and over again.
And not just any 2 letters. It happens at a very specific address in the genome. The algorithm showed these 2 base pair deletions almost always happen at a TNT motif.
Okay, let's break down the TNT. That stands for a thymine followed by any random nucleotide followed by another thymine, TNT. And the 2 letters that get permanently deleted at that exact address are almost always a C and a T. So, for a quick mental image for you listening, the DNA literally gets blown up at the TNT.
I love that visual.
Yeah.
Because the signature is so incredibly specific, a 2 base pair CT deletion occurring at a TNT motif, we don't have to guess what caused it. We know exactly whose fingerprint that is.
Which brings us to our rogue enzyme, the primary suspect, TOP1, or topoisomerase 1. But TOP1 isn't supposed to be a killer, is it? I mean, it's actually an essential part of the cell's maintenance crew.
Oh, it is completely vital. Without TOP1, your cells literally couldn't function.
Think of your DNA like an incredibly long, twisted, old landline telephone cord.
Oh, good analogy.
Yeah, when the cell needs to read the DNA to make proteins, it has to pull the strands apart. But if you pull apart a tightly twisted cord, the tension builds up ahead of you until the whole thing just knots up entirely.
Right, it physically can't unwind anymore.
So TOP1 is the enzyme that acts like a pair of microscopic scissors. It intentionally cuts one strand of the DNA, lets the tension spin out and untangle, and then it glues the strand perfectly back together so the cell can keep reading.
It relieves topological tension. But TOP1 also moonlights. It has a secondary backup job involving a process called RER, or ribonucleotide excision repair.
Okay, here's where the mechanism gets really fascinating. Your DNA is made of building blocks called dNTPs. But sometimes, especially when a neuron is stressed out or working in overdrive, the cell accidentally grabs the wrong building block, an RNA building block, an RNTP, and shoves it into the DNA strand.
It's like building a brick wall and accidentally slotting in a block of wood. It makes the whole structure completely unstable.
Right.
Normally, the cell has a dedicated repair crew for this exact mistake. An enzyme called RNase-H2 comes in, spots the wooden block, and gracefully removes it.
But the researchers found that in these diseased neurons, RNase-H2 is missing in action, like it's deficient. So that wooden block is just sitting in the DNA, and the primary repair crew is nowhere to be found.
Which forces TOP1 to step in as the backup crew. TOP1 arrives, uses its scissors, and tries to cut that wrong RNA letter out. And, under normal circumstances, it could handle the job. But remember that SBSB signature we talked about earlier, the massive oxidative stress.
Resisting from the inside out, yeah.
That oxidative stress physically damages the DNA right around that error. So when TOP1 cuts the strand to remove the wrong letter, it encounters that oxidative damage, and the damage acts like superglue.
Oh, wow.
It traps the TOP1 enzyme onto the DNA strand. It physically cannot detach. This creates a highly toxic roadblock called a TOP1CC, a TOP1 cleavage complex.
So the enzyme trying to fix the tangle gets permanently glued to the trap.
It is stuck. And when the cell's machinery tries to read the DNA and hits that trapped enzyme, the whole process violently aborts. The cell eventually has to rip the stuck enzyme off the track just to survive.
And let me guess, when TOP1 is violently removed, it leaves behind a scar.
Exactly. It leaves a permanent genetic scar. That specific 2 base pair deletion, our IDA signature.
I mean, think about how hard your brain cells work just to keep you functioning. If the repair crew gets glued to the highway every single time there is a pothole, the whole system just collapses.
It's a complete disaster for the cell.
Let's do another quick recap of the logic here before we look at the physical proof. We've established that the missing DNA is a highly specific 2 base pair deletion at TNT motifs. We know this specific genetic scar is caused by TOP1. And we know that TOP1 is getting permanently trapped on the DNA because of a lethal combination of severe oxidative stress and a missing primary repair crew.
That's the mechanism, yes.
But that is all based on algorithms and mutational signatures. If I'm a skeptic, I'm asking, how do you physically prove this invisible enzyme is getting stuck in dead brain tissue?
The researchers anticipated that exact skepticism. They didn't just rely on sequencing algorithms, they built a 3-tiered physical case. Tier 1 was proving the enzyme was actually stuck at the scene of the crime. To do this, they used an assay called RADAR,
which stands for Rapid Approach to DNA Adduct Recovery. How does RADAR actually pull that off?
Think of it like a highly aggressive chemical wash. The RADAR assay strips away all the loose proteins, all the normal cellular material, and only isolates proteins that have formed a covalent permanent bond with the DNA itself.
Oh, clever.
Yeah, and when they ran the diseased neurons through this wash, they found that TOP1CC, the trapped, glued-down version of the enzyme, was physically and massively elevated in the ALS, FTD, and AD brains compared to healthy controls.
So Tier 1 proves the enzyme is physically trapped. But does that trapped enzyme actually shatter the genome? That brings us to Tier 2 of the evidence, S1 nucleus gel electrophoresis.
Right. This step tests for the physical fragmentation of the DNA ladder. When TOP1 gets trapped, it leaves what's called a single-strand break, an SSB. One side of the double helix ladder is cut.
Just a nick on one side.
Exactly. Now, if enough of these individual cuts accumulate along the genome, they can overlap and become a double-strand break, a DSB. That is where the entire ladder snaps in half.
And to visualize this, they add S1 nuclease. That is an enzyme that specifically hunts down exposed single strands of DNA, those SSBs, and it cuts the opposite strand, forcibly turning a temporary nick into a catastrophic double-strand break.
Then they take that DNA and run it through a gel using an electrical current. Intact, healthy DNA is a large, heavy molecule. It stays neatly at the top of the gel.
Oh, okay.
But if the genome is shattered into thousands of tiny pieces by these breaks, those small fragments run all the way down the gel. Visually, it looks like a blurry smear.
And when they looked at the DNA from the diseased brains, it showed up as a massive smear. The genome was heavily fragmented, and crucially, the intensity of that smear perfectly correlated with how many of those two base pair deletions a patient had.
Which leads perfectly to the final, most advanced layer of proof, Tier 3. Because a skeptic might still argue that standard sequencing sometimes can't tell the difference between a temporary nick on one strand of DNA and a permanent deletion across both strands.
Right. If you just read a tiny fragment, you might mistake a repair in progress for a permanent deletion. So how do you definitively tell them apart?
They used a highly specialized technique called meta-CS.
Multiplexed end-tagging amplification of complementary strands.
Yeah, it is a type of duplex sequencing. DNA has two complementary strands, often referred to as the Watson strand and the Crick strand.
Right.
Standard sequencing usually mixes the signals. Meta-CS tags the Watson strand and the Crick strand individually with unique barcodes before amplifying them.
So they can look at the Watson strand and the Crick strand entirely separately. And by doing that, they can see if the two letters are missing from both sides of the ladder, proving it's a permanent deletion and not just some transient repair step.
Exactly. This duplex sequencing definitively mapped the chronological sequence of the cell death. It proved that the single-strand 2BP deletions mediated by TOP1 were heavily enriched in the diseased brains, and that those exact deletions were the direct precursor to the double-strand breaks that shattered the genome.
Man, it's an airtight molecular prosecution. But, in science, you always have to anticipate the ultimate alibi.
Always.
Someone is going to look at this paper and say, wait a minute, these are postmortem brains. The patients died, the tissue sat around, maybe the DNA just naturally degraded into a smear because it's literally rotting tissue. How do you know this trapped TOP1 mechanism actually caused the disease while they were alive?
To rule out postmortem decay, the researchers needed an internal control. They needed tissue from the exact same diseased brain, subjected to the exact same postmortem conditions, but from an area that doesn't get affected by the disease.
Right.
So they looked at the cerebellum.
The cerebellum is that dense structure at the lower back of the brain that coordinates balance and movement.
And remarkably, the cerebellum is generally spared in ALS, FTD, and Alzheimer's disease. The neurons there simply don't die off the way the neurons in the frontal cortex or motor cortex do.
So they isolated cerebellar granule cells from those very same ALS, FTD, and AD brains, and they ran them through the exact same gauntlet. The single-cell sequencing, the radar assay, the S1 nucleus gels.
And the result was zero.
Wow.
No IDA signatures, no trapped TOP1 enzymes, no smearing on the gel. The DNA in the cerebellum of these exact same patients was perfectly healthy and intact.
That is the definitive proof. I mean, it proves that this TOP1-mediated damage is not a postmortem artifact. It is a highly specific, targeted pathological process happening only in the vulnerable cortical neurons that define these devastating diseases.
When you pull back and look at the sheer scale of this evidence, from single-cell sequencing to duplex tracking of the Watson and Crick strands, it forces a massive paradigm shift in how we think about neurodegeneration.
Just think about what this means for you or anyone you know who has been touched by these diseases. For decades, billions of dollars in drug trials for Alzheimer's and ALS have focused almost exclusively on clearing out the protein garbage.
Yeah, trying to scrub away the amyloid plaques or dissolve the tau tangles or clear the TDP-43.
We have been obsessing over treating the trash rather than looking at the broken trash compactor.
This deep dive reveals a unifying mechanism sitting underneath all of those different proteins. The true shared driver of cell death is genomic instability, driven by oxidative stress and trapped TOP1 enzymes.
Which means the real therapeutic target of the future might not be protein-clearing drugs at all, The future of treating ALS or Alzheimer's might be drugs that prevent TOP1 from getting trapped on the DNA in the first place.
Right, or drugs that boost the cell's RER repair activity, or simply therapies that supply the missing RNase H2 so TOP1 never even has to step in and act as a dangerous backup.
We are moving toward therapies focused on stabilizing the very DNA of the brain, stopping the fragmentation before the neurons ever reach that point of no return.
It's incredibly hopeful research.
It really is. And I want to leave you with a final provocative thought to mull over. We've seen today how this exact same trapped TOP1 mechanism bridges completely different diseases like ALS, FTD, and Alzheimer's. But what about other brain diseases? Could conditions like Huntington's disease or Parkinson's or even severe forms of normal cognitive decline just be different flavors of this exact same DNA repair crew getting glued to the tracks? Does every human brain eventually succumb to this enzyme trap if it just lives long enough?
It really forces us to ask fundamental questions about the limits of how long our neurons are designed to live and whether this oxidative trap is the ultimate ticking clock for all human cognition.
Our diagnostic X-ray machine might have been broken, but it looks like we've finally found a powerful new lens to look through. Keep untangling those threads, and we'll see you in the next deep dive.